Today we will discuss the organelle genomes part 2. In the previous sections or the slides of this topic, organelle genomes, we have discussed about the different organelles which have the genomes and the discovery of the organelle genomes, the mode of the replications, transcriptions and the regulations of the transcription and the translations in the organelle genomes. In this topic, we will discuss and understand the significance of the study of the organelle genomes and their importance in the research and the treatment of the different uncurable diseases. The mitochondrial inheritance. In most multicellular organisms, the mitochondrial DNA is inherited from the mother and it is known as the maternally inherited. In the diagram, you can see the mode of the inheritance. The maternal inheritance means at the fertilizations, all the mitochondria are contributed by the oocytes, and the paternal transmissions of the mitochondrial DNA was reported in the skeletal muscles in a patient with a mitochondrial myopathy, and this was reported by the Schwartz and the Wishing in 2002. The mitochondrial inheritance mechanisms and conclusion is that the simple dilutions means an egg which have around the 2 lakhs mitochondrial DNA molecules, whereas a healthy human sperm has been reported to contains only the 5 average molecules. The degradations of the sperm mitochondrial DNA in the male genital tract and in the fertilized egg, and at least the few of the organisms and the failure of the sperm mitochondrial DNA enter the egg. Now the female inheritance. In the sexual reproductions, the mitochondria are normally inherited exclusively from the mother. The mitochondria in the mammalian sperm are usually destroyed by the egg cell after the fertilizations. Also, the mitochondria are only in the sperm tail, which is used for propelling the sperm cells, and sometimes the tail is lost during the fertilization process. In 1999, it was found that the paternal sperm mitochondria, which have the mitochondrial DNA, are marked with the ubiquitin to select them for later destructions inside the embryo, and some in vitro fertilization techniques, particularly injecting into the oocytes, and may interfere with this. The fact that the mitochondrial DNA is maternally inherited and enables the genealogical researches to trace the maternal lineage far back in the times. This is usually accomplished on the human mitochondrial DNA by sequencing the hypervariable control regions and represented as the HPR1 or HPR2, and sometimes the complete molecules of the mitochondria DNA as a genealogical DNA test. The HPR1 consists about the 440 base pairs. These base pairs are compared to the same regions of other individuals. Most often, the comparison is made with the revised sequences. Bill et al. Have published in his studies and tracing the maternal descent of the domestic dogs from the bulls. The concept of the mitochondrial Eve is based on the same type of the analysis and attempting to discover the origin of the humanity by tracking the lineage. The mitochondrial bottleneck. Now, The entity is subject to the uniparental inheritance and with the little to the no recombinations may be expected to be subject to Muller's ratchet. The accumulations of the deleterious mutations until functionality is the lost and the animal populations of the mitochondria avoid this through a developmental process which is generally known as the mitochondrial DNA bottleneck. The bottleneck exploits the random process in the cell and increase the cell-to-cell variability in mutant load as an organisms develop. A single egg cell with the some proportions of the mutant mitochondrial DNA thus produce an embryo in which the different mutant loads are the present. Cell level selections may then act to remove those cells with more mutant mitochondrial DNA and leading to a stabilized or reduced in mutant load between the generation to generations. The mechanisms underlying the bottleneck is debated with a recent mathematical and experimental study and provide the evidence for the combinations of the random partitions of mitochondrial DNA. Now the male inheritancy. Male mitochondrial DNA inheritance has been discovered by the Plymouth Rock chickens. The evidence supports the rare instances of the male mitochondrial inheritance in some mammals as well as. Specifically documented occurrence exists for the mice where the male inherited mitochondria were subsequently rejected. It also found that in the sheep and in the some cloned cattle and the rare cases of the male mitochondrial inheritance have been found in the humans. Although many of these cases evolved in the clones, embryos, or subsequently rejections of the paternal mitochondria, other document in the vivo inheritance and persistence under the lab conditions. Doubly uniparental inheritance of mitochondrial DNA is observed and in bivalve mollusk. In those species, the female have only one type of the mitochondrial DNA, whereas the males have the F-type mitochondrial DNA in their somatic cells, but M-type of the mitochondrial DNA in the germline cells. The paternally inherited mitochondria have additionally been reported in the some insects such as the fruit flies, honeybees, and some cycads. Now the mitochondrial donations. In IVF means in vitro fertilization technique known as mitochondrial donations or mitochondrial replacement therapy represent as the MRT, which results in offspring containing the mitochondrial DNA from a donor female and a nuclear DNA from the mother and the father. In the spindle transfer procedure, the nucleus of an egg is inserted into the cytoplasm of an egg from a donor female which has had its the nucleus removed but still contains the donor female's mitochondrial DNA. The composite egg is then fertilized with the male sperms. The procedure is used when a woman with the genetically defective mitochondria wishes to procreate and produce the offspring with healthy mitochondria. The first known child to be born as a result of the mitochondrial donations was a boy, born to the Jordanian couples in the Mexico on 6 April 2016. Now the susceptibility of the mitochondrial DNA. The concept that mitochondrial DNA is particularly susceptible to the reactive oxygen species, represented as the ROS. It is generated by the respiratory chain due to its proximity, which remains the controversial. The mitochondrial DNA does not accumulate any more oxidative base damage than the nuclear DNA. It has been also found that at least some types of oxidative DNA damage are repaired more efficiently in the mitochondria than in the nucleus. The mitochondrial DNA is packed with the proteins which appear to be as the protective as present in the nuclear chromatin. Now the next is the mutations and the diseases. Moreover, the mitochondria which evolved a unique mechanisms which maintains the mitochondrial DNA integrity through the degradations of excessively damaged genomes followed by the replications of intact repaired mitochondrial DNA. This mechanism is not present in the nucleus and is enabled by the multiple copies of the mitochondrial DNA which is present in the mitochondria. The outcome of the mutations in the mitochondrial DNA may be in the alterations in the coding instructions for some proteins, which may have an effect on the organism's metabolism or maybe their fitness. Now the genetic illness, which is related to the mitochondrial DNA. The mutations of the mitochondrial DNA can lead to a number of the illness, including the exercise intolerance and the Kearns-Share syndromes represented as the KSS, which causes a person to lose the full functions of the heart, eye, and muscle movement, like the paralysis. Some evidence suggests that they might be major contributors to the aging process and age-associated pathologies. Particularly in the context of the disease, the proportions of the mutant mitochondrial DNA molecules in a cell is termed the heteroplasmy. In the diagrams, you can see the heteroplasmy. For heteroplasmic mitochondrial DNA mutations, the cell can compensate for the reduced wild-type mitochondrial DNA until a certain threshold is met. The functions of the cell become compromised in this case, and threshold depends on the specific mutations and the types of the cells. Examples, the neurons which have a lower threshold for the disease state. Within cell and between these cell distributions of heteroplasmy dictate the onset and severity of the diseases and are influenced by the complicated processes within the cell and during the development. Mutations in the mitochondrial transfer RNAs can be responsible for the severe diseases, and the mutations in the nuclear genes that encode the proteins that mitochondria use can also contribute to the mitochondrial diseases. These diseases do not follow the mitochondrial inheritance patterns, which is based follow and the Mendelian inheritance patterns. Now, the use of the mitochondrial DNA in the disease diagnosis. Recently, mutations in mitochondrial DNA has been used to help the diagnosis prostate cancer in the patients with negative prostate biopsy. The mitochondrial DNA alterations can be detected in the biofluids of the patients with with the cancers.